Hello, everybody. This is Miss Peachy from your WCA Biology A class. And today we are doing a little overview of Unit 5, Lesson 2 in your Biology A class. So, this lesson is part of the unit on mitosis. So, we're starting a new unit today. And today we're talking about complex organisms and kind of how they function. And we're kind of starting to talk a little bit about mitosis itself, the process of mitosis. So as always, I do have a little lesson kind of outlined for you to follow. So that is on my website. It is linked right underneath the video for today. And if you open that up, you can kind of follow along and take notes in here. Split your screen, take notes on it or print it out and write on it so that you have the appropriate notes for today's lesson. All right, so, we start off with this lesson kind of talking about how something as simple as a fertilized egg becomes an entire organism. How does this happen? And the big thing is through the process of mitosis, a cell can make copies of itself. And then through cell differentiation, those cells can turn into multiple different kinds of cells that can build an entire organism. So let's kind of remind ourselves a little bit here about kind of the structure, I guess, of what's going on inside your cells, what's going on inside your DNA. Okay, so I'm going to just open up my whiteboard here. And actually, I'm just going to quick open up a new whiteboard for us. And so we're just going to remind ourselves of what's kind of going on inside of our DNA. I'm going to see if I can get these whiteboard tools. Actually, that's fine. We'll just use the ones we have. And I'll move this aside. And just remember that in the nucleus of your cells, where your DNA is housed, we are eukaryotic organisms. So we have a membrane-bound nucleus, is one of the organelles inside the cell. So you have your cell, and inside of the nucleus, which is bound by its own little nuclear membrane, which I'll try to draw with like a really thin line here. Not very good. We have our DNA, which I've been playing with how to draw this better because my pictures are so terrible. It's getting a little better. There we go. And as you can imagine, that's just one tiny little piece of a strand of DNA. Again, in human beings, we have 46 chromosomes. So there's 23 pairs of chromosomes, and each one of them has a very long strand of DNA that's all kind of wound around these histone proteins and kind of coiled up so that it can all fit nicely and compacted inside of that nucleus. In fact, your lesson talks about how long your DNA actually is. It says if you were to unwind all of the DNA in one cell of your body, it would be about 6 feet long. So about the height of a grown adult man is about how long the DNA is in just one cell of your body. Okay, remember you've got billions of cells in your body because you're kind of a big person, and cells are very, very small, right? So within each strand of DNA, if we zoom in a little bit here, right? Pretend like we're getting in there real tight. We're just like in a microscope, and we're able to kind of just get really in here. Then you've got these little segments that all kind of code for a specific thing. And those little segments that are found in the DNA are called genes. And genes are just like a portion of the code that is all kind of coding for a specific characteristic, a specific trait, or a specific job that your body is doing, right? And so within your DNA, you've got these areas which, again, are genes that code for things. And then you've got areas that really don't code for anything, or we don't know maybe what they code for, but they don't seem to be part of genes. So there's parts that are actually specifically designated as genes, and then there's other just kind of filler code in between as well. All right, so DNA and genes, right? So again, the DNA then is all kind of coiled up together and around these histone proteins, and it forms kind of these long compacted structures that we call chromosomes, right? And they've got, they're kind of joined together there with what we call the centromere. Oops. I just Okay. And so I wanted to put a couple of pictures in here. So I just kind of pause it for a second to get those pictures in place. But so when you have the chromosome, remember that you have two copies of each chromosome. So I'm going to zoom in on this thing. This is called a karyotype. And your karyotype kind of shows you your chromosomes. And it's kind of cool because it can kind of show you a picture of your chromosomes. So here's all the way number 1, number 2, number 3, number 4. It's a little blurry as I zoomed in here, but there are 22 up to number 22, and then that last set, remember, is the XY set that determines sex. If you're XY, this person is a male. If it's XX, that person would be a father, a female. XX would be a female. Okay, I'm just reading the word father here. That's why that's. So as you can see here, this is color coded on this karyotype. The purpley color is from mother. The green color is from father. So we kind of see that little X shape that we normally associate there with it. But what we're looking at here are two different strands, one inherited from your mother and one inherited from your father, right? So when we look at that kind of little structure that we would normally see, it is half from mother, half from father. Okay, so that's what our chromosomes are. Then we also have a term called a chromatid. And a chromosome, a chromatid, they kind of get a little bit confusing sometimes. But like here again, it would be like the mother, father, right? During the process of replication, so that's part of the cell cycle, which we'll review again in a moment. Remember that part of the cell cycle during the S phase, the synthesis phase of the cell cycle, the DNA is going to make copies of itself so that it is able to pass on a copy to the new cell. When it replicates itself, now we have these sister chromatids. And so basically, the blue makes a copy of itself. So now there's two blues, and it's joined to. We get that centromere that joins together. And the red makes a copy of itself. So we have two reds, right? So now we see that familiar X-like structure, and that only forms prior to mitosis during that synthesis part of the cell cycle. Otherwise, they kind of are, they're individual strands like this. We don't get the familiar X-like structure like we see over here until we get replication that happens to kind of prepare the cell for cell division. So now we've got, basically, we are doubling our DNA. At that point, remember, here we would have 23 pairs. As you can imagine, right before mitosis, you're going to end up having each one of these is going to be doubled. So then you'd have two of these guys here, right? So every one of them would look like a little X rather than just an individual line. And then they'll separate and then they'll divide, and that's what happens during mitosis. So I just wanted to make sure you're familiar with kind of the different terminology here. That when we see that familiar X-like structure, that is just prior to cell division. Otherwise, what you are normally is just 23 pairs of individual chromosomes. Okay, so then going back to the lesson here, going to minimize this here. You can see these pictures here. It talks about mitosis, and it talks about DNA replication that happens during the cell cycle. And if we remember during the cell cycle, and actually let me just pull up that picture real quick of the cell cycle as well, because we did this in the previous lesson. Okay, so during the cell cycle, which we have here, right? I'm actually gonna kind of split my screen here if I can, just to make this. Come on. There we go. So we can both kind of see both screens here for a moment. We actually have DNA replication that happens, and that happens in this part. That's the S phase. So each of the 46 chromosomes is duplicated by the cell. That's happening during interphase. And interphase is basically all the blue and this little bit of pinkish red color. Everything except for mitosis is considered interphase. Okay. So again, we talked about this again in the last couple of lessons here. The first step of interphase is the G1 phase. This is basically the first gap or the gap 1 phase. And this is where the cell is growing. Sometimes we call this the growth phase, the G standing for growth. But the cell is growing here. It's duplicating everything in the cell except for the chromosomes. So all the organelles are being duplicated. The amount of cytoplasm is being increased to be able to make a larger cell. And all that is happening in the G1 phase. And then our S phase is when we have replication of the DNA. So the DNA unzips and a new strand of DNA is made, 2 new strands where once there was just one. And then we have the G2 phase where we have double checking. We have more growth that occurs as well. We also have those 3 kind of checkpoints as well here that occur kind of, in 3 different locations along our cell cycle. So again, with that S phase, it says during this new DNA is synthesized and chromosomes are copied into 2 identical sister chromatids. And that's kind of what we saw here, right? So these chromosomes copy themselves. They're copied, and now there are 2 of each copy. We call them sister chromatids, and that's where that familiar X kind of shape comes from. And then the third step of interphase is known as the G2 or second gap, duplication of molecules, replenishing of energy, and creating the proteins needed for mitosis. Okay, so all that's kind of review, except for maybe the part about the sister chromatids and the differences between a chromosome and a chromatid. So now we get to the point of mitosis. And I actually think that the lesson does a pretty good job here, giving a lot of details, but making it understandable and kind of good pictures as well. Not everything in the lessons I think is amazing, but some things are really good. And this is one of the things that I think is really good. So mitosis itself is the process by which the cell, then this much larger cell with lots of organelles in it and lots of chromosomes, is now going to divide and make 2 cells. And it's really important that all that stuff in interphase we talked about actually happens, right? Because if it didn't happen, then if the cell stayed the same size and then divided, we would have 2 little cells instead of 1 regular size cell, right? So if the cell can get bigger first, and then each of that divide, then we end up with 2 of the same size cells instead of 2 smaller cells. We want to maintain the same size, because otherwise you can imagine if that keeps dividing, then eventually your cell's just going to disappear. And you don't want your cells to be too small, because then they can't accommodate all the stuff that needs to be packed inside of them. You also want to make sure that you conserve the number of chromosomes. If you have 46 chromosomes, you don't want to divide and end up with 23 and then divide and somehow get 12 in one and 13 in the other, or something like that, because that wouldn't make sense either. Or 12 in one, 11 in the other. That wouldn't make sense either. So you have to double the number of chromosomes first so that you still maintain the exact same cell after cell division happens. So what's cool here, I think, is it talks about kind of the different stages, but it tells you what's happening in each of the stages. So I'm actually going to bring this up in kind of center stage here. So it talks about the formation of some really cool structures in the cell called the mitotic spindle and the microtubules. So these are structures made of protein structures that help to kind of mechanically guide all the stuff around in the cell. The mitotic spindle is kind of the attachment point, and then the microtubules will attach to the mitotic spindle. And then these microtubules come down from either pole of the cell, and they act as fingers and they grab stuff. They actually pull the organelles around, they pull the chromosomes around, and they pull the cell apart. They're kind of mechanically there as like little robot hands to kind of move things around inside the cell. And I think that's actually kind of a cool thing. So our first stage of cell division, first stage of mitosis, is known as prophase. And in prophase, what happens? So let's look at it. The cell begins to prepare for replication by reducing the size of the chromosomes. Before this, the chromosomes were way more spread out. And then they start to shorten up and thicken. And actually, that X-like structure that we're familiar with kind of finalizes its shape at this point. So again, you want to make them shorter and smaller and more compact so that when they move around, they don't get tangled up with each other. It's really kind of practical. The reason why it happens is so like these long, like if it's really long and spindly like spaghetti or really long hair, it's going to get all tangled together. And that would be no good. You're going to end up with lots of breaking and mutation and stuff like that. So if you can get them really short and thick, they'll more easily be able to move around inside the cell without getting broken, without getting tangled. So that happens in early prophase. The mitotic spindle, a small structure within the cell that will help organize the chromosomes, are formed. So that's on the ends, remember, of the cell. And then in middle prophase, the spindle keeps growing, and then it develops those finger-like projections, the microtubules, that act like, it says long arms, I say fingers, that start to capture and organize the chromosomes. And later on in prophase, one microtubule from each side of the spindle attaches to capture every chromosome. So kind of like the crane game that you play. But as you can see here, we actually have now each arm coming down and attaching itself to a individual chromosome. Okay. It does not say this in here, but what also happens in prophase is the nuclear envelope is going to dissolve itself. And that actually happens pretty early on as well. And I think it's kind of an important point. This nuclear envelope here has to dissolve itself before those microtubules can attach to the chromosomes. Otherwise, it would be in the way. So that's really kind of an important point as well that's not mentioned in this step-by-step kind of guide. The next stage is metaphase. And metaphase, I always remember, metaphase means in the middle. It's the middle phase, but it also physically the chromosomes get lined up in the middle. So the chromosomes here are lined up in the middle of the cell so that they are easily able then to be directed later, where half of them will move to one side and half of them will move to the other side. So as you can see here, remember, these are those X-like structures, right? And then they separate and half will move to the top of the cell and half moves to the bottom of the cell. This is anaphase. So if we go to our picture here, now remember we started with this, you have this. So now what happens then during this separation is one of these guys will end up going to the top of the cell and the other one will go to the bottom of the cell. And that happens with every chromosome. So your new cells will end up with basically this situation right here, right? Each one of them will have one of the blues and one of the reds instead of two of the blues and two of the reds, or two blues, or one cell gets two blue and one cell gets two reds. You don't want that. You want to have everybody having exactly the same stuff. That's kind of the whole point here. Okay, so that is anaphase. Microtubules pull the lined up chromosomes apart, separating them into sister chromatids. Each pair of the chromatids moves towards opposite ends of the cell. This ensures new cells that form, called daughter cells, will each have chromosomes. Will have each chromosome, one of each kind. And then telophase, two new nuclei form. So now you have, at this point, There's the picture. At this point, you've got these moving to opposite ends of the cells, right? But now you have the nuclei that are actually going to form. So I don't like this picture very much. But if you can remember what had initially happened, is that nuclear um, envelope dissolved itself. Well, it has to reform. And it does this. And so now we have this situation. Let me just get rid of this DNA here. We have this situation here where we have, say, our chromosomes at either end of the cell, right? So then a new nuclear envelope is going to form around those chromosomes. So that's what's going to happen during our telophase or telophase. I always said telophase, but then I hear other people say telophase. So I don't know. All right, and then finally, cytokinesis. The cytoplasm divides, forming two new chromosomes. So what you see here then is what's happening is, oh, I don't want that, is the cytoplasm now starts to divide. Picture that as like almost like a figure eight or something. The cell kind of starts to pinch off, the cytoplasm divides, and then two new cells form, where once there was just one. And during this whole process, I mean, they kind of oversimplified a little bit, but remember, you've got those you got those spindles, and you've got those microtubules and stuff. They're all kind of directing what's going on, like those little fingers. They're pulling it apart. They're also coming down and grabbing organelles and putting the organelles where they need to go so that every cell that forms is exactly the same as the other cell. So the end result of mitosis is cloning the cell. You're exactly making a duplicate of it, of like a photocopy. Nothing is different. All right. Why would you need to do this? Because stuff happens, right? I mean, you hurt yourself and you scrape your hand and it has to heal. And in order to heal, it has to make new skin cells to cover that wound. You're growing taller. The only way you can grow bones and skin and muscle is by mitotic division, mitosis. So growth and healing and some cells just replace themselves because they get damaged through contact with lots of things. Like your skin is constantly replacing itself, like I mentioned to you the other day. So all of these things are happening because of the process of mitosis. But mitosis isn't where it always ends. So remember, your cells in different parts of your body are all very different from each other. Your cells that make up your skin are different from the cells that make up your heart, that are different from the cells that make up your brain. And so those cell types occur because of something called cell differentiation. So differentiation just because becoming something different, right? Pretty simple. I think that word is pretty simple. And cells that differentiate are known as stem cells. And stem cells, there are actually multiple different kinds of stem cells. There are stem cells that happen to occur only in embryos. And those are embryonic stem cells. There's two different types of embryonic stem cells. There's totipotent and pluripotent stem cells, which are both embryonic. So let's put those on our little note sheet here, huh? So I'll just write them down. Zoom, zoom, zoom. Here we go. So these are the first stem cells that embryos make, or the first stem cells we see in stem cells, or in embryos, prior to them even being considered an embryo at that point. So these can become any other type of cell, and they also turn into the placenta. And since the placenta is not inside the embryo's body, it's attached to the uterine wall and it ends up kind of interfacing with the mother bloodstream to be able to provide nutrients and things, and oxygen to the placenta or to the embryo and be able to kind of rid waste products from the embryo as well. It's kind of called like it's not part of the embryo's body itself. So that's why only totipotent cells can actually do that. The other one is pluripotent cells. So these are the second type of embryonic stem cell. They appear second. And they can also turn into any other type of body cell, but they cannot turn into placental cells. They're a little bit more limited, but they're still pretty pretty flexible as far as what type of cell they can become. And then we have what are known as adult stem cells, or we call them multipotent stem cells. These are ones that you find in not in embryos. They can become many different types of cells, but they are limited. And generally, you have specific types of multipotent stem cells. Like you might have a certain type of stem cell that is specific to bones. Um, and so they only can become bone cells, right? Or different types of bone cells. Um, I think they're called oogenetic cells or oogenic cells. That's what they're called. It's like a bone stem cell in a human. It's called a oogenic cell, and they can become osteocytes, which are like just general bone cells. They can become well, they actually they turn into osteoblasts first, and then osteoblasts can become osteoclasts or osteocytes. So, but they're limited to just kind of a different variety of bone cell. They can't become any type of cell in the body. And so you've got these specific adult stem cells that are already differentiated to a degree, and they can become kind of something else within a small range, and they occur in multiple places of your body. Okay. So there's a lot of controversy about using embryonic stem cells because embryonic stem cells have like the unique ability to become any other type of cell, right? So they're kind of cool in that regard to be able to use them to regenerate maybe body parts that are broken and things like that. Um, However, obviously, the ethical dilemma here is that you're taking them from an unborn baby. So it's an embryo. You can get some, I think you can get some pluripotent stem cells from like umbilical cords, but you're still limited in like what you can harvest in that way. The cool thing is that there's really cool research. It's fairly new in genetics that's called induced pluripotency. And they've actually found that they can take just regular old multipotent stem cells and they can um, do something. It's some sort of enzymes that they apply to it that actually reverses kind of their development and they can become pluripotent again, which I think is kind of amazing. So they could take your stem cells from your skin and they can induce pluripotency into them and they can become these pluripotent stem cells and then those can then be made into anything else. And they've actually used this type of technology to be able to develop I think they're still pretty much in like the research stages, but I know they can develop things like little pieces of skin and stuff for skin grafting, and they've actually used them to build like miniature hearts and things. But it's really again kind of in its infancy as far as what they're using it for. but it's kind of a cool, like, cutting edge technology when it comes to treatment of disease using kind of biotechnology here. I'm not sure what you'd even call it. So it's pretty cool. Anyways, that was not mentioned in the lesson either, so I thought I would point that one out. All right, so that's pretty much it for what was in the lesson, ladies and gentlemen. And so I will see you in the next lesson.